We're going to ask our presenters now to become panelists, and we will be able to entertain your questions. Please, Amy and Gaurav and Sean. As we're assembling, just a time check, we are going to break promptly at 10 o'clock, so we have about 22 minutes for discussion. The intention here is not only to answer specifically questions that you may have of any one of the presenters, but also to try and seek for the information of the National Academy where you see the future of what we've just heard today ought go. In other words, if you have any futuristic thoughts of needs which we may not be able here to answer or address, should they be addressed in the future? So if you would please, let's see, Chris, you're already there. So if there are two microphones, Rich, you'll be next. That'll take about 20 minutes. This will take about 20 minutes. That'll be the end of the session. Go ahead, Chris.
I'll be quick, but I'm Chris
Marshall
from Pfizer, but I do have two quick questions. The first is, and I'll start with the safety aspect using the alectinib, ceritinib example you've given. How do you incorporate into real-world data or real-world evidence being used for regulatory approvals or for regulatory submissions the safety aspect? So, for example, alectinib versus ceritinib, perhaps it was better, the overall survival, but the safety signal was not. Was that incorporated into that submission?
Oops. Great question. So I was sitting here when I was listening to Sean speaking. One of the things I was thinking a lot about was specifically about safety. And there are a couple of different issues. So as you think about generating large data sets at scale, I'm going to come to this written alectinib in just a second. If you think about generating data sets at scale, thinking about how we generate safety data at scale is something that is a real problem to be tackled. And it comes in several categories. So some data, that safety data at scale, I think we already have. So information, for example, laboratory information such as hepatic and renal data, we've got that, right? The second category is safety data that requires the interrelationship of multiple data points and how they come together. So febrile neutropenia, which needs us to think about fever, evidence of neutropenia, and potentially hospitalization, right? Or something else that signifies clinical consequence, that actually requires us to think about an amalgamation. And then the last is that other types of information needs to come directly from the patient. So how do you incorporate, for example, patient-reported outcomes into safety data at scale? So I think we have a data task to solve for that. That's one. It's much different when you have a very carefully pre-specified question, and you can go and look at least what is available for the patients within the context of that question. We did not do it in the alectinib ceritinib example, as far as I know, but we have a number of examples where then we can go and look in the chart and say, for this population of 500 patients for each group, what do we understand has happened in terms of, for example, development of cough and shortness of breath, or development of evidence of febrile neutropenia. So there you can actually curate, but you actually have to pre-specify exactly what adverse events you're looking for. Does that help?
Thank you. That's helpful.
Did that help, Chris?
That is.
A shout out to you, Chris, and Pfizer in general, because it was they who originally brought in a request to Project Datasphere to convene the immune-related adverse events workshop last December that really brought forward the importance of understanding myocarditis in the instance of checkpoint inhibitors that brought the other families in. So again, thank you and Mace and Patrick Cobell, the father of TAPR and 1,000 patients on trial.
Rich Shilsky from ASCO. So I'll just pick up on the same theme for a minute. And it really relates to the whole issue of how to understand attribution in adverse event reporting, particularly in real-world data, right? So we've all looked at lots of MedWatch reports, lots of electronic medical records. It's relatively easy to identify when an adverse event, meaning something bad has happened to a patient. Whether that has anything to do with the treatment administered or not is another issue. So I'm wondering, Sean and Amy in particular, how you've begun to think about how to resolve this issue of attribution in evaluating real-world data.
Sean?
Well, that's a very actually complex topic. Even when we look at, for example, the audit that we did, part of the reason that only 14% of the reports coming to the FDA are uninformative is because of attribution, that it's very hard. In the United States, it's the companies, the sponsors' responsibility to make the attribution. And typically, it's a very complex exercise, especially if you're looking at each case in a silo. And since there are limited data assets at the IND stage of clinical development, attribution is really a subjective exercise for the most part, and it's in the eye of the beholder. Outside the U.S., specifically EMA, but other regulatory territories, attribution is really tied to the investigator's assessment, but in the United States, it's tied to the sponsor's assessment. So there are those differences that also make it even harder to make the appropriate assessment when it comes and distributing these reports globally, because the majority of clinical trials are currently global, and there are these different definitions and requirements, regulatory requirements when it comes to making that attribution. However, once you have a data set, and if the idea of global central bank scales, and we don't see any major barriers in terms of its scaling, because the value proposition, again, is very clear to sponsors and also to regulatory agencies, we can start to look at it analytically. And we can start to incorporate algorithmic methods and more predictive methods of helping either the sponsor or the investigator to make that attribution. Because right now, as we all know, we're looking at narratives and MedWatch forms, and it remains a subjective way of looking at attribution.
Amy?
In addition to what Sean said, I've been trying to push us to go in 4 different directions. There's what we're doing right now, which is when there's something that looks like an adverse event, we can look into the chart and say, what did the physician say was going on? And so we've got a number of things that we're working on specifically, where we're looking to say, what did the physician say is going on here? What did he or she believe? And what kind of decisions did they make based on that? What did they do with the drug? How did they treat the patient, et cetera? So there's kind of case review and that approach. The second, and this goes back to my sort of point of adverse events at scale across data sets, is to be able to essentially get background rates that allow you then to watch for changes as compared to background rates. And that's where I'm hoping that the real world data and sort of Flatiron goes, is kind of getting us to a place of having enough continuously common adverse event monitoring that we can then use that as a background rate. The third place that I think we don't talk about enough is to use the solutions, the software solutions that are already in our clinical workflow. So right now we've built in adverse event monitoring into our electronic health record. And so we're basically, the physicians now have that as a part of their documentation option. And so in that, there's also the option of being able to describe attribution. So there's, from that point of view, where it's passive, but then there's also an active opportunity in that continuously monitoring against some kind of background rate or background standard. If you see something pop up at an individual patient level, going back to the physician saying, what did you see going on here? Because we know who the physicians are. We actually are already in their clinical workflow and can develop those tools. And I think that's where a lot of the space is going to end up going. That makes sense?
Thank you. Let's see, I believe it. Yeah, please.
Hi. 2 questions, both about data that you didn't show, but it sort of begged. It was evident that you could easily show that. First, for Gaurav, did you look at PD-L1 as a function of time in terms of its predictiveness, like you did for the continuous value of TMB?
Gaurav.
Yeah, so happy to speak to that, and we're writing some of this up as well. But basically, we recapitulated some of the findings we saw yesterday, where PD-L1 independently predictive, as well, independent of TMB in terms of just interrelationship, but superimposed with TMB seems to play less of a contributory role than TMB.
And second, for Amy, clearly you had all those drugs there. Sort of the elephant in the room is, do you try to compare them? That is, what if you look at the PD-L1 binders versus the PD-1 binders, or is this politically forbidden?
Oh, it's politically very doable. And yes, we look at drug classes, we look at individual drugs, we look at individual drugs as it relates to, for example, the setting of PD-L1 at different expression thresholds. So indeed, we're able to do that. People who are using our data sets are able to do that. Most importantly, a lot of these questions, others who are using our data sets, especially our academic collaborators, tend to be the ones who are looking at and asking some of the comparative effectiveness questions in particular. So the answer is very much so, yes.
Emmett.
You notice I didn't give you the answer.
Emmett.
First, we should make sure that the attribution for that slide probably goes to David on the tumor mutational burden. Is that correct? Okay, just so that we clarify the footnotes.
The royalty payments will be sent, yeah.
Slightly pointed question for Amy, which she'll get where I'm going, and a comment for Sean. So you gave us a lot of OS data and you made claims, and yet you also told us that you had a flaw in your OS data, which is missing data, and yet you gave us no confidence intervals. And the FDA correctly holds us accountable for every single OS point. And we can't make a claim without every single OS point. And I learned from a master, Roger Dancy, how you do that. So by the way, we can probably tell you how it's done if you have to literally get every OS point. And yet, can you really make the claims you made without confidence intervals?
So for the purposes of the pictures, the confidence intervals weren't there, but the confidence intervals are sort of at the bottom of the figures. Important point that you just brought up. And so I glossed over the issue of mortality data, but I cannot begin to talk about how important this is. So I want to hit on this in three different ways because I think you're pointing at some
We
important
have
things.
to be brief, though, because we have at least 5 or 6 other questions in as many minutes.
I promise. One is that there is not a national data resource for mortality data, so that you have to amalgamate it across multiple sources. And the second is that you need a way to benchmark sensitivity and specificity so that you understand how that data are performing for specific examples, so that you understand how much missingness there is and how it's interrelated to the problem at hand. And then the third is that you should be able to also essentially do simulations to see what happens when mortality data has higher levels of sensitivity and specificity, and look at the impact on the overall results. We have a paper in Health Services Research about this from 2 months ago, including the calculation of sensitivity and specificity for those data sets. So at all times, I think that's a characteristic you have to include when you look at this, including then impact on confidence intervals.
Yeah, just quick response to that. I mean, there ought to be a threshold at which you don't report the data because there's too much missing data.
Totally agree. That's one of the things we proposed.
And then for Sean, just an out-of-the-box thought. I mean, one of the things that I've learned as a clinical monitor with 2 first-in-human phase 1 studies is exactly that. I mean, signal finding when you have all the intent in the world to protect a human being is very hard because the investigators actually don't know what it looks like on our side. They have a threshold of saying it might be, so I'm going to attribute it. And despite the FDA's best efforts to re-educate, that still is a phenomenon happening. I just wonder if at some point we ought to rotate through each other's worlds. If the academics should rotate through Merck and Merck should rotate through the FDA. I say that somewhat in fun, but just to say that we don't all understand what this looks like from each other's side. And I think it's really important for this really critical thing of patients volunteering their lives to us.
Bravo. Sean, do you have any comment on that? I know Percori has some experience there, and of course, you're entrepreneur in residence, although it doesn't address Emmett's question directly or suggestion. Please.
Right, absolutely. So we have part of what we're doing through these collaborative arrangements is basically disseminating our institutional knowledge to stakeholders, both academic and industry partners, but also draw from their experience and incorporate the learnings into our regulatory framework. And especially in today's world where you need to have an agile mechanism when it comes to data science, science and technology, and clinical trials are becoming very much tech and data science oriented. You need to have a more collaborative, agile approach while maintaining, obviously, the authority that the FDA has to protect the patient's safety and protect the public. So we firmly believe that by looking at data rather than subjective metrics and narratives, we can start to create transparency and also harmonize and looking at safety information across clinical studies. So I think it all has to do with removing subjectivity and judgment and moving more towards analyzing data and having an analytical approach with confidence intervals and uncertainty measures when we look at safety and also efficacy. And actually ties in, that also relates to the question that you asked, that obviously in real-world data repositories, there may be missing data. However, if you look at clinical trials, especially in adult oncology, we're only capturing the experience of 3% of patients. And the majority of cancer patients, adult patients, are excluded from clinical trials. So the effect size estimate that we get with the p-values and the confidence intervals are very volatile when that drug is then given to all patients in the post-market setting. So there's volatility when you look at traditional clinical trials. That estimation of the treatment effect is not actually the benchmark. If you think about, in most cases, where 97% of that patient population was not included in the exercise of estimating that treatment effect. So you could expect, so that is probably in a lot of cases, in adult oncology especially, not the appropriate benchmark, because if we replicate the results of traditional clinical studies, that's great using real-world data. We know that high-quality data can do that, but we can also go beyond that and capture the experience of folks that are not typically represented in clinical trials. And some of that data was presented earlier by Amy, patients with organ dysfunction, brain metastasis, HIV, history of autoimmune disease, elderly, minorities. These are patients that are typically not in traditional clinical trials.
Thanks, Sean. It should be noted that in the terms of the global safety, which you addressed during your presentation, recently Dr. Pastor just returned from a trip to China, where we have on good authority, because I got plenty of spies there, was greeted as a rock star, rightfully so, and who had many discussions on this on a global scale across many different families of drugs. So that's also being addressed in a global sense. I think we have a question here, and then we'll go over to the next slide, please.
Hi, Mark Stewart, Friends of Cancer Research. I think my questions may be a little more forward-looking, I think, and shifting from the drug to the diagnostics, and curious what the role or opportunity there is to learn from real-world evidence, particularly as I think much of our knowledge or understanding of patient selection based off of PD-1 and other biomarkers that are coming to fruition are focused on monotherapies, and as this meeting's highlighted, there's much work being done in the combination space. So as these are being used in the clinical setting, are the appropriate data elements and meaningful information being collected in EHR that could support the clinical validity of these tests in different combination settings? And more maybe from a diagnostic standpoint and regulatory perspective, is there an opportunity for real-world evidence to support the modifications to tests, particularly around next-gen sequencing, where you might have 3 or 4 that are companion diagnostic, but clearly there's hundreds of gene variants, and maybe at the time of approval there wasn't enough information available, but over time you're able to collect that.
If you go on much longer, you'll have to show a slide. That's all there's to it. It's a great series of questions. Gaurav, would you like to try and knock that one out?
Sure, I'll try to tackle it. So on the first point, I think that the methods we've described here could extend to combination therapies, sort of similar to how they would extend to monotherapies. And if you have the same endpoints and you have the same diagnostics, and the regimen, I think, is sort of extendable. Your second question, I think, is extremely interesting, which is how could real-world evidence impact the idea of diagnostics and companion diagnostics or complementary diagnostics? There are a couple things that I think are happening semi-contemporaneously, which enable that to, I think, be a really exciting part of our future. So one is the FDA has been sort of very creative and sort of adaptive in how they thought about the notion of approving diagnostics, and obviously Foundation Medicine was part of that story, but the notion that diagnostics can be approved as a platform with sort of a rigorous methodology for revision and modification is, I think, a fundamentally different way and sort of progressive way of thinking about a diagnostic sort of universal backbone. As you know, the Foundation One CDX test that we've recently launched is FDA approved, and that backbone is approved. So one of the things that becomes, I think, really interesting is if you combine real world use of an FDA approved backbone with now real world sort of regulatory grade endpoints, and you learn, for example, a novel biomarker that stratifies response to a therapy that exists on that backbone, the path to sort of regulatory optimization of diagnostic to therapeutic association, I think, becomes potentially much shorter. So that's, I think, we are just on the verge of being able to do that for the confluence of reasons that I just described. So I'm very excited about that future.
That was a great discussion, Gaurav. Thank you very much. From Case Western Reserve.
Thanks. Stan Gerson from the Case Comprehensive Cancer Center. I gave Amy 2 hours to prepare her response to this question we walked over this morning. So I'm sort of forward-looking, if I could, about how real-world data will help us manage case report forms and manage the issue that Sean really brought up, which is the other 97%, and whether and what the milestones would be for shortening drug review and approval with confidence in real-world data.
My comment will be quick. I think data quality, at the end of the day, it has to do with having access to high-quality data that's captured at the point of routine care. And the FDA has a mechanism to verifying data integrity and quality through primarily source document verification and inspections. The Office of Scientific Investigations is the body at the FDA that validates data integrity and assesses data quality. And if we have access to high quality real world data, then we can start to make those assessments appropriately.
And I think that follows on with, therefore, we can't fall down on the task of getting the data as clean as possible and curating it with as much detail as possible. The other part that we didn't talk about is the ability to trace back to the source. So when data points are curated, they're coming from some source documentation, often what the clinician is seeing and writing down. And so when needed, to be able to get back to what actually happened and be able to have confidence in how those decisions were made to go from source document to a curated data point that you're using in very high-risk questions. I think the other point that you said was about case report forms and filling things in. And as we imagine the case report forms in the past, we would fill in 45 pages because we were afraid that if we didn't fill in everything right now, we were going to lose the chance to fill in the information. And I think in the future, there's more opportunity for parsimony. You clean up or curate 2 pages worth, and then you can go back and fill in the rest of the information if you need in the future.
It's emblematic of this particular panel that we have still questions that remain to be asked. Unfortunately, we're going to have a break now in which you'll be able to ask them individually. We've been treated from the 21st Century Cures Act and its importance to the FDA, to sponsors, and most importantly to patients. We've also been treated to the fact that there are a couple in this room that arrived with the FDA getting messages by carrier pigeon, and then the great leap of faith into the fax machine, which is still the era in which we're in. But there's an architect in the room by the name of Sean, with a patron by the name of Rick and Scott, that have taken now us to an informed level. And the opportunities there are incredible for sponsors, both for-profit and not-for-profit. And we've also been really given a glimpse inside 2 entities that, although started separately, recognized that coming together there would be synergies. And that was Foundation Medicine and Flatiron. And in the midst of all of that, the real recognition was seen by a major oncology sponsor saying this is where this entity must go on behalf of not just the company and for drugs, most importantly for patients. So we started at the beginning with patients. Let us end there also, that all of this big data all of these numbers, all of these clinical trials and real-world data, all are patients with the tears removed. Thank you for our panelists, and thank you for great questions. Thank you, Academy.